All right, let's take a look at translation in a little bit more detail for HL. The first thing we're going to look at is the initiation complex for translation. So we know translation's occurring at a ribosome, and there's a few more details about the ribosome that you're going to need to know. The first of which that it's made up of two subunits. There's a large subunit and a small ribosomal subunit. So here's the large and here's the small. To initiate translation, we have the mRNA has to dock on the small ribosomal subunit where the first start codon, which is AUG, is in the proper place on the small ribosomal subunit. And then the tRNA that is the complement to that, so it's anticodon there for the AUG codon. The anticodon is UAC, and it is going to be carrying the methionine amino acid. So the start codon being AUG, and that codes for methionine. So once these three players get together, the small ribosomal subunit, the mRNA at the appropriate place with the start codon in position, and then the initiator tRNA that carries the methionine, once these three are together, then the large ribosomal subunit will come in and attach. And that's the translation initiation complex. Okay, so now translation can start, but we're going to do a little bit more detail here with the with the ribosome. So this is just another kind of three-dimensional graphic of a ribosome. So here's the large and small subunit, and you can see that there are these three sites, these three areas in the ribosome where the tRNA molecules will attach on this large subunit. I'm going to show you a cartoon version of it because it's a little bit easier. Okay, so here's the ribosome and its cartoon version. The mRNA binds to the small subunit, and then we've got the large subunit that has three binding sites. Okay, the P, this P site, it's called, it stands for peptidyl tRNA binding site, but I just think about it as the position one with a growing polypeptide. So the P one is kind of the center one, the one that's going to be getting everything started. And we'll see that the first tRNA is going to be positioned here with the start codon. And so the P is going to be holding the tRNA that actually has that growing polypeptide chain. So you can see here it's in the P site, and this is the one that has the growing polypeptide chain, so this chain of amino acids. The A site is where the next tRNA is going to come dock. So it's the amino acyl tRNA binding site. You don't need to know the technical names of these, just the A site is fine. So in the A site, the next tRNA is going to dock. And so now you see the P, it's holding the polypeptide chain, and then the A site is right next to it. So these two amino acids, the polypeptide chain and the amino acid of the tRNA on the A site are going to then be catalyzed. That peptide bond is going to be catalyzed here. Okay. Once that happens and it's catalyzed, then translocation happens, and then this tRNA will move over to the E site. I call it the exit site. And actually they do too. So then that's going to move it over, and then that's the exit site. It doesn't have any more amino acids associated with it or a polypeptide chain, so it'll move here and then can just leave. Okay, so remembering it in order, I think in general, if you remember that the P site is the one that holds the polypeptide chain, and then maybe alphabetically you've got the A site and the E, or you could read it backwards as APE, or so there's a few different ways. So if you can try to remember the order of which they're in is important, but just remember that E is exit, P is polypeptide, and A is the first one on the list. Okay, now we know that synthesis of the polypeptide, like we did last time in 2.7, it's these repeated cycles. Now when you're going through and doing it, you need to think about the sites and where they are. So here's our P site, right, with the polypeptide. And then that's when we get the codon recognition coming, where the A site and the new tRNA will dock. And then we get the peptide bond formation happening. And then everything shifts over and we get translocation happening. So that moves over, it goes to the E site and can exit. So when you're talking about the elongation of a polypeptide, so elongation in translation, then you need to be talking about the positions on the ribosome, including the EP and A sites, okay? And then lastly, we get the termination of translation, which is when we get the release factor coming in. And then instead of course, a tRNA, it's just going to be a protein that can then help disassemble all the parts together when that release factor binds. So when you're breaking down translation, just like in transcription, talk about initiation. So you'll be back here with the initiation complex, right? So talk about initiation and all the different things that are important here. We've got the assembly of the translation initiation complex and all those parts. Then you'll talk about elongation, which involves, the AP and E sites and the, those three parts: codon recognition, peptide bond formation, and translocation, always making sure you pay attention to those ribosomal docking points for the tRNA. And then lastly, termination. So initiation, elongation, and termination. Okay, so make it, when you're trying to sequence your way through it, if you just kind of put those in order, you should be just fine. Okay, another important thing that for HL that for sure they might ask you is where the ribosomes are depending on the fate of the protein. So free ribosomes, just ones that are freely in the cytoplasm, those are for use primarily within the cell. Possible enzymes that are used inside the cell that are necessary for, assembly of things, or, maybe it's a polymerase or something like that. So any proteins to be used within the cell are on free ribosomes is where that happens. But protein synthesis for proteins that are going to be exported from the cell or secreted, and that would be done on the rough endoplasmic reticulum, because we know the rough endoplasmic reticulum is studded with ribosomes. So if you're a pancreatic cell and you're making a bunch of insulin, that's going to be on the rough endoplasmic reticulum, where that insulin will then be packaged and then sent to the Golgi and then secreted out of the cell. There's a really nice animation that I've shared with you guys that you can take a look at there too. One thing that's interesting is in prokaryotes, because they do not have a nucleus, is that prokaryotic cells can start translation while transcription's still happening. So you notice we've got RNA polymerase, so transcription's happening, meaning we're going from DNA to RNA to mRNA. But notice there's all of these ribosomes attached to that mRNA, even though, but there's no nucleus. So you don't have to wait for that mRNA to come out, and it doesn't do mRNA processing in prokaryotes and bacteria and things, and it doesn't have to go to the cytoplasm because it's already there. So translation can occur, and you can actually see that happening. Notice that here is the DNA in a prokaryote, and there's a whole bunch of these ribosomes attached. So you've got the DNA that you can see here, and then all the ribosomes attached to it from there. And we can actually call these polysomes, is a name for them. So one after the other. So this is kind of showing you these strings. They look like a bunch of beads hanging off the side of the DNA. Okay. And so, but another thing that's interesting with multiple ribosomes can be on a single mRNA in the cytoplasm of a eukaryotic cell. So if they're hanging off in strings coming off of a DNA, so you can see there's like an anchoring string there, this is going to be a prokaryotic cell. But if you just see an mRNA with a whole bunch of ribosomes hanging off of it, and then it's got a bunch of, you can actually even see those polypeptides being formed. This could be in a eukaryote, okay? Because you can have a whole bunch of ribosomes that are translating one mRNA at the same time. It doesn't have to wait for one to finish. I think one of the last things we're going to touch on here is the fact that tRNA, we know that after it leaves the E site, right, here's the anticodon on the tRNA, it needs to bind with its proper amino acid. And when it does bind with its proper amino acid, there's actually specific enzymes that do that. And so each tRNA will have a specific enzyme associated with it that will only attach the appropriate amino acid to it. And these enzymes are called tRNA synthetase enzymes. The longer version is the amino acyl, but you could just go with tRNA synthetase enzymes. And they will take a specific tRNA that's got that, whatever the anticodon is there, and will attach the appropriate amino acid to that one. And this will demonstrate, this actually is a good demonstration of enzyme substrate specificity, because if you attach the wrong amino acid, then the genetic code that we know wouldn't work the way it does. So that's a really important factor. And I think the very last thing I want to touch on in this video is that you should be able to recognize a tRNA in in a micrograph. And well, it's not really a micrograph, but using molecular visualization software. So this is kind of what tRNA looks like if you look at it with molecular visualization software, and you should be able to identify the part where the anticodon is and the part where the amino acid attachment site would be. So this is what the tRNA looks like. This is a cartoon model of it where the anticodon and then the attachment site. So you should recognize that. And you also need to be able to recognize kind of a ribosome, which for me, I don't really, I'll be honest with you, I don't always technically know when and how. The only difference that it looks like a giant mass of a ball, but you might be able to see the 3 binding sites if there's tRNAs in there. So you can see there's 3 tRNAs that have these binding sites. And without the tRNAs, it would be really hard to see what those were. So just be aware that there could be a picture of a ribosome, but you for sure, I have seen them ask you questions that you should be able to recognize a tRNA.